In part 2 of this chapter, we're going to look at the special senses, which include the sense of smell, touch, taste, vision, hearing, and equilibrium. We'll begin with this video discussing the two chemoreceptors. Those are the sense of smell and the sense of taste, or olfaction and gustation. So olfaction is the detection of odorants, chemical molecules dissolved in the air. Replacement and sensitivity of receptors tends to decline as we get older. It becomes more difficult to smell things that don't have a very strong odor to them. So odorants are simply molecules that are dissolved in the air, and then they bind to chemoreceptors in the nasal mucosa. So this provides information about food, people, danger, even just flowers. We can distinguish between thousands of different odors. So the olfactory epithelium is the sensory receptor location and where the receptors are housed in that epithelial layer. So it's located in the superior part of the nasal cavity, and it has three different types of cells. You have the receptor cells that detect odorants. You have the supporting cells that basically are the supporting cast, like we saw with the cells in the brain, the glial cells. So your supporting cells, of course, are going to be those that help the receptor cells, and then your basal cells are constantly replacing the receptors. All right, let's take a look at this image here. Now, what you see, of course, here is the olfactory, the nasal cavity lined with that mucous membrane. Here is your olfactory nerve. Do you remember what nerve that is? Cranial nerve number 1. Yeah, if you didn't remember, you could read, right? So your cranial nerve number 1 extends out toward the anterior aspect of the brain. Now you can see these little fibers sticking down through this bony area. We can look at it in enlarged view here. So this bone, remember, is which one? That's part of the ethmoid bone. Remember, because the little holes that are in the ethmoid bone are there in the cribriform plate of the ethmoid. And that's what these olfactory nerve fibers extend down through from the olfactory nerve down into the olfactory epithelium. And those, of course, are your receptors. Now, the primary neurons in the sensory pathway are those olfactory receptors. They have a bipolar cell structure. Remember, that means two extensions off the cell body. So you have a single dendrite and an unmyelinated axon. At the distal end, you have cilia that project from that dendrite. And this is where the chemoreceptors that detect the odorant are located. Now, the perceived smell depends on which cells are stimulated. So your olfactory nerve, remember, is cranial nerve number 1, and it's a bundle of olfactory axons that extend through the cribriform plate of the ethmoid bone and then enter right back into the olfactory nerve. So those olfactory bulbs, okay, you see here sticking out toward the anterior aspect of the brain, are located just under the frontal lobe. And the olfactory nerve fibers are going to synapse there with mitral and tufted cells. So these are just part of the olfactory nerve and the associated structures. So this forms connections with the olfactory glomeruli. So the olfactory tracts are actually paired, and that's bundles of axons of these cells on the inferior frontal lobe surface. They project directly to the cortex, the olfactory cortex that's located in the temporal lobe. And in addition to that, the information is always also sent to the hypothalamus, the amygdala, and other brain regions. But as we talked about when we learned brain regions, we do not send these nerve pathways through the thalamus like the other sensory pathways go. So with all that said, how do we smell? Well, we have the odorant binding to the mucous membrane. Okay, so there are proteins in there that bind to that. The olfactory sensations begin when that odorant binds to the protein, and the protein then stimulates the olfactory receptors, which are rapidly adapting. Remember, that's why we don't smell that candle or Uncle Joe's stinky cologne for a long time. That then, of course, is going to send to a secondary neuron that conducts the signals to the central nervous system areas for processing. So the cerebral cortex says, oh, there's a smell. It's apple pie. The hypothalamus has your visceral reaction to that, and it's like, oh, every time I smell apple pie, I think of my grandmother because she always made that for me. And in addition to that, the amygdala ties the emotional aspect to it. The hypothalamus gives us that visceral reaction. So your salivary glands start to secrete and you start getting ready to eat that pie. And the amygdala, of course, in addition to adding the emotional reaction, also helps us to recognize what that smell is. Our second special sense, also involving chemoreceptors, is gustation, or the sense of taste. So this is the detection of tastants. So we have gustatory cells that are chemoreceptors located within taste buds. When you look at the surface of the tongue, you see all these little projections called papillae. We actually have three different types of these. The filiform, as you see here in the first little image, are very short and spiky. They don't have a role in taste. They simply help us to move food around, to manipulate it in the mouth and across the surface of the tongue. The fungiform papillae are mushroom-shaped. They contain a few taste buds, and they're located primarily on the tip and the sides of the tongue. And then we have the foliate papillae. So your foliate papillae have sort of leaf-like ridges. They're not very well developed. They have a few taste buds early in childhood, and they're located primarily on the posterior lateral aspect. And then we have one more, which are the valate or circumvallate papillae. These are the largest, and we have the most of these. And that's a good thing, because that's where most of our taste buds are located, if you see here on the lateral aspect of those papillae. So they are located in rows of about 10 to 12 all along the posterior dorsal aspect of the tongue. Your taste buds themselves are sort of shaped like an onion, and they house the receptors. So we have gustatory cells. Those are the receptor cells that detect the tastants. The supporting cells basically sustain the gustatory cells, help deliver nutrients, and help to get rid of old, broken down, worn out cells. And then our basal cells, well, think of those as your stem cells. They replace the gustatory cells as they are worn out, or maybe we damage them by drinking or eating something hot. We got to replace them. So your gustatory cells are neuroepithelial chemoreceptive cells. So neuroepithelial, nerve and epithelium, chemoreceptive, detect chemicals in liquids such as saliva. So your tastants are the tasty molecules that are in our food, and they get dissolved in the saliva and they stimulate the microvillus. The gustatory pathway, so we've got the receptors, the chemoreceptors located on the papillae of the tongue. So they detect that sensation and they project it to the medulla oblongata in the brain stem. In the anterior parts of the tongue, the sensory neurons are part of facial nerve number 7, or cranial nerve number 7, which is your facial nerve, and the posterior 2/3 are sensory neurons that are part of the glossopharyngeal nerve, which is cranial nerve number 9. So the secondary medullary neurons project that information through the thalamus. Remember, the thalamus is our relay center for the special senses, except for olfaction. Then we have tertiary or third thalamic neurons that send that information to the gustatory cortex located in the insula, which is that deep lobe within the brain, for processing. Absolutely. And that is where we detect and determine what we're tasting. So we have 5 basic taste sensations. Sweet is produced by organic compounds such as sugar or artificial sweeteners, so they stimulate your sweet taste buds. Your salty taste sensations are produced by metal ions like sodium and potassium. Sour is associated with acids such as citric acid or vinegar. Bitter taste buds are stimulated by alkaloids, such as we find in unsweetened chocolate. However, a lot of toxins and spoiled foods also have a bitter component to their taste, and that's supposed to be protective for us so that we don't keep something that could be toxic in our mouth and then swallow it and it gets into the body. And our 5th type of taste buds are umami, and these taste buds are stimulated by savory or meaty flavors such as amino acids and monosodium glutamate, which we usually just call MSG. So again, the gustatory pathway, the primary signal comes from the receptors in the tongue. So the medullary activity is going to trigger salivation, so we start filling the mouth with saliva. If it is something that perhaps has spoiled, we'll get a nauseating stimulus, and instead of the ooh, let's eat stimulus, and that can trigger us to gag or vomit so that we get rid of whatever it is that stimulated those taste buds. Now, let's assume it is something good. So we've tasted apple pie or whatever it is that you're thinking about, and that, of course, sends the information to the medullary area associated with taste, which is then going to send that information straight up to the thalamus, and the thalamus says, okay, that's taste, let's send it to the gustatory complex, which is in the anterior aspect of the insula, and that is where we determine what it is we taste. Now, taste is more than just that chemoreceptor. It also involves texture and temperature, but even more so the sense of smell. So food has less taste if olfaction is blocked. For instance, you've got a cold and your nose is all stopped up. Well, those odorants that are the chemicals that are part of your food, we detect those on chemoreceptors in the tongue, but some of them actually waft up into the nasal cavity and get detected as odorants. And that is why we have a much better sense of taste when we can smell, because the two work synergistically.